Today I am going to deal with the last part of the chapter biomolecules proposed by NCERT, NTA for the NEET examination. Okay, so today I am going to deal with enzymes. So what is actually an enzyme? An enzyme is a protein that is speed up thermodynamically possible chemical reactions without changing the equilibrium constant or delta G. That means enzyme is a protein and it speed up some chemical reactions, and that chemical reaction is a biochemical reaction. So when it speed up the biochemical reactions, it cannot use it up and cannot change the equilibrium constant. Okay, so when the enzymatic reaction taken place, whatever the amount of the enzyme used, that much enzyme you will get after the reaction completed. Okay, so enzymatic reaction, enzymes cannot use it up. It can use many times for the same reaction to take place. Okay, so there are some nucleic acid which function as an enzyme. Okay, for example, ribozyme. It is actually the RNA that function as an enzyme. That means all the enzymes are not proteins. There are some exemptions to that. That is some of the nucleic acid. For example, RNA that acting as an enzyme. For example, ribozyme is an enzyme. At the same time, it is a nucleic acid. Okay, an enzyme, like any protein, that has a primary structure, it also have secondary structure and also have tertiary structure because it is a protein. Okay, when it shows a tertiary structure, it folded and crisscross itself and produce many crevices, or it produce many small pockets. One such pocket is called active site. The significance of this active site is the pocket into which the substrate coming to fit for the chemical reaction or the enzymatic reaction to be taken place. The substrate coming to attach on this active site. So active site is the pocket on which the substrate is attaching. Substrate means the substance or the chemical on which the enzymes acting, that is known as a substrate. Okay, so during the enzymatic reaction there are enzymes that acting on a particular chemical substance, that is known as a substrate. Okay, thus an enzyme at their active site catalyze reactions at a high rate. Okay, if the substrate attaching to any other place, enzymatic reaction or the chemical reaction cannot speed up. So when it attaching to the particular pocket, that is active site, then only the enzymatic reaction speed or enzyme speed up the chemical reaction. Otherwise, it cannot participate in the reaction. Okay, so enzyme catalyst differ from the inorganic catalyst. You know that there are so many catalysts you studied in the chemistry also, that is the inorganic chemistry. So this inorganic catalyst is also catalyst, but it is different from the enzyme. Differences between enzymes, that is a biocatalyst, and inorganic catalyst. So what are the differences? One of the differences is that all enzymes are proteinaceous in nature. We already told, except a few, that is the ribozyme. But inorganic catalysts are actually inorganic in nature. Okay, so enzymes are organic, proteinaceous in nature. Inorganic catalysts are inorganic in nature. That is one difference. Another difference, enzymes are quite large as compared to the size of the substrate. So enzymes are large, but the substrate are always small. Okay, but in the case of the inorganic catalyst, there is no difference in the size of catalyst and the substrate. Both are maybe similar or it may be different. Okay, another difference, the enzymes are complex macromolecules with a 3D structure, but that is not in the case of the inorganic molecule. Inorganic catalyst are simple molecule. It has no 3D structure. For example, magnesium ion is acting as a catalyst. It is not a macromolecule and has no 3D structure. Okay, another difference, enzymes have high molecular weight. Okay, but inorganic catalyst have low molecular weight. Again, example, magnesium ions or copper ions. Another difference, the enzymes are originated in biological world, living system, and that mediate biochemical reactions. But an inorganic catalyst that speed up the chemical reactions, it is not originated from the biological world, and it is not catalyzing the biochemical reactions. Okay. Another difference, enzymes are highly efficient. So it actually it speed up 10 raised to 10 times the chemical reaction speed up, biochemical reactions, but that much is not in the case of the inorganic catalyst. Okay, so enzymes, another difference, enzymes are specific reactions. It is very highly specific. One enzyme catalyst only a particular substrate, but that is not the case in the inorganic catalyst. It is not highly specific. Okay, then another difference, in the case of the enzymes, activity regulated by specific substance, but in the case of the inorganic catalyst, that is not at all regulated. Okay, then the another difference, in the case of the enzymes, enzyme effective in narrow range of the temperature and pH. Okay, but in the case of the inorganic catalyst, it is not regulated by narrow range of temperature and narrow range of the pH. Definitely temperature regulating, inorganic, and pH regulating, but that is not the narrow range. Okay, then you see inactivated enzymes are protein. I already told enzymes are proteins, so these enzymes are inactivated at a low temperature. For example, at 10 degrees centigrade and below, enzymes become inactive. But when the temperature increases, the enzymes again become active. At the same time, enzymes are protein, so at a high temperature, the enzymes are undergoing denaturation. And the denatured enzymes cannot regain again into the enzyme activity when the temperature decreases. Okay. However, still enzymes are isolated from organisms which normally live at a high temperature. For example, hot vents and sulfur springs. Some bacteria living in the hot vent. Vent means the deep part of the ocean where continuous volcanic eruptions and earthquake taken place, so the temperature is very high. Some of the bacteria also living in that temperature, and that having enzyme, and these enzymes are working at high temperature. And a similar case in the sulfur spring, the temperature is very high, boiling point, near about the boiling point, still some bacteria present there, this bacteria living, and that also with the help of some enzymes. So in these cases, enzymes are working at a high temperature, near about 80 to 90 degree centigrade. Enzymes are working in these organisms. Okay, so thermal stability is thus an important quality of such enzymes from thermophilic organisms. The organism living in high temperature in such a condition is known as a thermophilic organisms, and these thermophilic organisms have high stable enzymes at a high temperature. So that is the difference between inorganic catalyst and organic catalyst. Some aspect of the chemical reaction. Okay, chemical compounds undergo two types of the changes. You know that studied in the chemistry, that is physical changes and chemical changes. So what is actually the difference between the physical changes and chemical changes? In the case of the physical changes, it refers to change in shape without breaking bonds. So bond cannot be break in the case of the physical change. Okay. Similarly, changes in physical state, but at the same time the physical state will change, but there is no breaking of the bond. For example, ice melt into water, so only change the state from solid to liquid, or when water become vapor, from the liquid to vapor, there is no chemical bond breaking the chemical bond, only the state change, only the shape change. So that type of the reaction is known as the physical changes. That type of the changes is known as the physical changes. Okay, then let us see what is the chemical changes. The chemical changes involves breaking bonds, and new bonds are formed during the transformation. So when one substance transforming into another, there is a breaking of the bond and creation of the new bond taken place. Such a type of the change is known as a chemical change. Okay. For example, if a barium hydroxide react with sulfuric acid, it produces a barium sulfate and water molecule. Definitely, the barium hydroxide, the bond broken, and similarly H2SO4, the bond is broken, and a new bond formed in the barium sulfate and H2O. It is an inorganic chemical reaction. Okay. Then let us take an organic chemical reaction. For example, starch hydrolyzes to glucose. Starch hydrolyzing with the help of the enzyme into glucose. So starch is a large, long molecule. It is broken into smaller pieces and converting into monomer glucose. So there is a breaking of the bond and finally it converting into monomer. And this type of the reaction, that is actually organic reaction. Okay. Then the rate of physical change or the chemical process, that refers to the amount of the product formed per unit time. Whether it is a physical change or chemical change, the rate means how much product is formed after the process, physical process or chemical process. Okay, it can be expressed in the form of the equation, that is the rate is equal to delta P upon delta T. P delta means a smaller quantity, so using the notation delta. P means a product, and again T means in a particular time. It may be second, minute or hour. Okay. Then the rate can also be called velocity. The rate also can be called velocity if direction is specified. So you studied well in the vector, in the case of the physics, that is the velocity. If velocity is specified, velocity, we can say the velocity of the reaction if direction is specified. Okay, the rate is influenced by factors like temperature. This rate influencing temperature also, and even the pH change also will affect this rate. Okay, the rate of chemical reaction, this rate or the chemical reactions rate double, or it may be decrease by half for every 10 degrees centigrade change in either direction. That is, when 10 degree increasing, the reaction rate will increase. 10 degree decrease, the rate will become half. So on either side. So that is the nature of the chemical reactions. That much is sufficient for this enzymatic reaction, study of this enzyme part. So now we can examine how do enzyme bring about a high rate of chemical reactions. I told enzyme is the biochemical catalyst which speed up chemical reactions, biochemical reactions. So how the enzyme bring about a high rate of chemical reactions. Okay, see here, the chemical which convert into a product is called substrate. Okay, the substrate is the substance on which enzyme acting, and that converting into product, that is known as the substrate. Okay, an enzyme which have active site, we already explained, that convert substrate into product. Okay, enzyme has an active site, substrate attaching to that active site, and this enzyme converting the substrate into product. That is, the substrate gives product. Okay, by enzymatic reaction. Okay, when substrate bind to its active site, there forms a complex. That complex is known as the enzyme substrate complex. That is a complex ES. Okay, that is enzyme plus substrate. It produces ES, enzyme substrate complex. This complex formation is a phenomena. It is a phenomena. Okay, hence a transition structure is formed between the enzyme plus substrate to enzyme substrate. There is a complex enzyme substrate complex. It's a transition structure that is formed in between that. Directly cannot forming the enzyme substrate. Between that there is a transition stage. Transition structure is formed. Okay, immediately by making and breaking bonds, the product is released. So after the formation of this transition structure, the chemical bond is breaking and then new bond is producing and the product is formed and that will release. Okay, from this active site. Between the enzyme substrate complex and the product, many unstable transient forms are there. So between the enzyme substrate between the enzyme and the enzyme substrate complex and the product, there are so many transient stages. So the substrate has to go through much higher energy state. The substrate has to go much energy state, higher energy state or transition state. This higher energy state is known as a transition state. Okay, and this transition state is well shown in the graph. Okay, the difference in average energy content of substrate form that of this transient state is called activation energy. The difference in the average energy content of this substrate form and this transition state is called what is activation energy. That activation energy is shown in the graph. Okay, enzyme eventually break down this energy barrier. Actually, enzyme, what will do enzyme break this energy barrier. Okay, making the transition of the substrate to product more easy. So what actually enzyme doing? Enzyme break this activation energy and reduce the activation energy. Okay, activation energy actually is the energy needed to start a biochemical reaction. See the graph. In this graph, it is well shown that without enzyme, the activation energy is high. And when enzyme is adding to the reaction, the activation energy is reduced. So reduction of the activation energy, that means it speed up the chemical reactions and the product is formed very soon or very easily. If without enzyme, it is requiring a lot of time because the activation energy is requiring high activation energy. To bring the high activation energy, it is requiring long time, but enzyme bring down this activation energy and the chemical reaction is speed up and the product forming immediately. Okay, so enzyme actually lower the activation energy, thus increase the rate of reaction. So enzyme plus substrate, it gives enzyme substrate complex, and this enzyme substrate complex is producing enzyme product complex, and then enzyme breaking out and it producing enzyme and product. Okay, and this enzyme again and again can be used. So we say that during this enzymatic reaction, enzyme is not used up, it only bring about decreasing the activation energy. Okay, we can study factors affecting enzyme activity, various factors affecting enzyme activity. We already explained that biocatalyst or enzyme are affected by various factors like temperature, pH, etc. But inorganic catalyst not affecting temperature, affecting temperature, but the narrow range is not there, and pH also affecting the inorganic, but it is not narrow. Okay, so what are the factors affecting the biochemical catalyst enzyme? One of the factor is the optimum temperature. Okay, so each enzyme shows its highest activity at a particular temperature. See this graph, it is showing the highest activity, showing the bell-shaped graph and showing the highest activity at this point. Okay, called this temperature is known as the optimum temperature. So we taken in this graph temperature on the x-axis and the enzyme activity on the y-axis. So highest activity taken place at this point, that temperature is known as the optimum temperature. Okay, activity decrease both above the temperature and below the temperature, optimum temperature. See here, below the temperature activity is less, below the temperature activity less, and high temperature also activity is less. Okay, so that temperature is known as the optimum temperature. And similarly, optimum pH is also there. Enzymes shows highest activity at a specific pH. So if you take y-axis and this is x-axis, on the x-axis we take the pH range 1, 2, 3, 4, etc. And on the y-axis enzymatic activity, you see the enzyme activity is obtained at a highest point here, and this is known as the pH optimum. Below the pH, the activity is decreased, and above the pH also activity is decreased. Okay, for example, in the case of the pepsin in stomach, the pH is 2. That means acidic. And similarly, duodenum, trypsin in the duodenum, it is requiring pH 8, that is alkaline. Okay, so most intracellular enzymes function best around the neutral pH, near about neutral pH. Most of the intracellular enzymes working. Okay, so that is the pH optimum. Then let us take a substrate concentration. What is the effect of the substrate concentration on the enzyme? See, when substrate concentration is increased, the reaction of enzymatic reaction also increase. If you take this graph, this is the y-axis enzymatic reaction, enzymatic activity, and on the x-axis you can take the substrate concentration. When the substrate increasing, the enzymatic activity also increased. Okay, when the reaction rate become maximum, that is known as the Vmax. This is the maximum reaction, that is known as the Vmax. No further, after that, no further increasing possible by adding the substrate. You add the substrate again and again, the enzymatic activity cannot increase. So the maximum activity took place at this point, that is known as the Vmax. Okay, further the reaction activity cannot increase by adding more and more substrate molecule. Why it happens? Because the enzyme having active sites, the substrate, all the substrate become engaged in this active site, so there is no active site remaining vacant. So further increasing the substrate cannot accommodate this substrate on this enzyme, and hence the enzyme activity cannot increase. And all this because the active site, all the active site accommodated by the substrate, further adding the substrate, this substrate cannot having place to attach on the substrate because all the active site is engaged with the substrate already. So this is the Vmax, and half of that is known as the half Vmax or 1/2 Vmax, and the when we plot on the x-axis that data we obtained, that is known as the Km value, Mendelian constant, that is the Km value. When the Km value is less, the enzyme is more active. When the Km value is progressing, increasing, the enzyme become weak and weaker. That we will study in the inhibition aspect of the enzyme. We are going to study the classification of the enzymes. Okay, see here, enzymes are classified into 6 major classes. So what are the 6 major classes? One of the class is the oxidoreductase. Number 2 is a transferase. Number 3rd one is a hydrolase. Number 4th one is a lyase, and number 5th one is isomerase, and the last 6th one is the ligase. Okay, all the enzymes falling under these 6 categories. Okay, so let us take first oxidoreductase. Oxidoreductase, it is a catalyst. The catalyst oxidation or reduction of the substrate. Okay, so enzyme which catalyze oxidation or reduction of the substrate, that is known as oxidoreductase. Example, if you take cytochrome oxidase, cytochrome oxidase actually catalyze the reaction alcohol plus NAD. In the presence of the alcoholic dehydrogenase, it produces aldehyde and NADH2. Here alcohol is NAD and alcohol that undergoing oxidation and reduction. Okay, and produce aldehyde and NADH2. NAD become reduced to NADH2 and alcohol become oxidized to aldehyde. Okay, so that is the oxidoreductase. Then if you take the second one, that is a transferase. Transfer a specific group from one substrate to another. So they taken a group from one substrate and it is giving to the another group and forming a new substance that is known as a transferase. For example, glutamate pyruvate transaminase. Glutamate pyruvate transaminase. See here, glucose plus ATP. That catalyzed by glucokinase and producing glucose 6-phosphate plus ADP. Okay, here the group transferred from the glucose and produced glucose 6-phosphate. So the one phosphate group is taken and giving to the glucose, and the glucose 6-phosphate is formed. Okay, then another one is the hydrolase. These break down large molecules into smaller one by addition of the water and catalyze breaking of specific covalent bond. Okay, most digestive enzymes belong to this category. Okay, for example, amylase, which hydrolase starch. So sucrose, example, in the presence of the sucrase is converting into glucose and fructose. Okay, by hydrolase mechanism. Okay, then fourth one is the lyase. See here, it catalyzes to cleavage specific covalent bonds and removal of group without hydrolysis, means without addition of water. Same reaction but without hydrolysis, it break the group and producing new product. Example, histidine decarboxylase, that cleave carbon-carbon bond in histidine to form carbon dioxide and histidine. Like this reaction, it produces XCC bond there, C bonded with X and another C bonded with Y, and it break into XY. It produces XY plus C double bond C, that is known as a lysis. Okay. Similarly, histidine decarboxylase to histamine and carbon dioxide. Okay. The fifth one is the isomerase. This catalyze the rearrangement of molecular structure to form isomer. That is isomer. Rearrangement producing the new structure, that is known as isomers. For example, phosphohexose isomerase, which change to fructose hexose isomerase. See here, glucose 6-phosphate is converting into fructose 6-phosphate. It is an isomer. Only the phosphate group taken from the sixth position, and then it produced a fructose 6-phosphate. So glucose 6-phosphate converted into fructose 6-phosphate. Glucose 6-phosphate converted into fructose 6-phosphate. Okay. Then the last one is the ligase. So what reaction it is catalyzing? See here, it catalyzes a covalent bond between two substrates to form a large molecule. Okay, the energy for this reaction is derived from ATP. Pyruvate carboxylase combines pyruvate and carbon dioxide to form oxaloacetate at the expense of the ATP. That is an example. That is the pyruvate carboxylase combined pyruvate and carbon dioxide to form oxaloacetate at the expense of the ATP. Okay, so pyruvate plus CO2 plus ATP, pyruvate carboxylase, it produces oxaloacetate plus ADP and plus inorganic phosphate. So these are the different classes, categories of the enzyme. That is the classification of the enzyme. Well, now we can study the mode of enzyme action, how the enzyme activity taken place. That is the mode of enzyme action. One of the theory proposed for this mode of enzyme action is lock and key hypothesis. Okay, this hypothesis was forwarded by Emil Fischer in the year 1894. You know, lock and key. One lock requiring one key to operate. If the key is different, the lock cannot be operated. So like that, one enzyme for the one substrate. Just like lock and key. Lock is large, key is smaller. So enzyme is representing the lock and key is representing the substrate. So enzyme is always larger than the substrate. Okay, so that we cannot study the in details of that. Another mechanism is the induced fit theory. So what is actually induced fit theory? Who proposed that? This theory was proposed by Koshland in 1959. It is proposed by Koshland in 1959. Okay, according to this theory, the active site of this enzyme contains two groups. So what are the two groups? One of the group is a buttressing group, and another one is the catalytic group. Okay, so what is actually buttressing group? Buttressing group is meant for supporting the substrate. It is giving support to the substrate when it attaching to the active site. And then catalysis is taken place, and the catalytic group is the group which is converting the substrate to product. Okay, so that is the induced fit theory. Nothing, it enzyme have two parts. That is a buttressing group that is helping to attach the substrate to the enzyme, and a catalytic group that is converting this attached substrate to the product. Okay, so that is the two mechanism of the action of the enzyme. Then let us study the inhibition of the enzyme activity. The inhibition of the enzyme activity means the enzyme activity become reduced by combining with some substrate or by adding some substance into the enzymatic reaction. Then the enzymatic reaction rate will decrease. So that substrate is known as the inhibitors. Okay, so let us see how the enzyme inhibition taken place. There are two types of the inhibition, that is the competitive inhibition and a non-competitive inhibition. So what is the difference between competitive inhibition and non-competitive inhibition? In the case of the competitive inhibition, some substrate that is very closely resemble the substrate. So the substrate and the substance that we are adding, that is very closely related molecules, substance, and this this substance that inhibit the enzyme action, then it is called a competitive inhibition. So some substrate closely resemble the substrate, inhibit the enzyme action, is called competitive inhibition. The competitive inhibitor is called the inhibitor is called the substance we are adding, that is called inhibitor. This inhibitor is called a competitive inhibitor. Okay, the competitive inhibitor bind to the active site, and the enzyme cannot catalyze the change of substrate into product. Okay, hence enzyme action declines. Okay, example for that, inhibition of the succinate dehydrogenase enzyme by malonate, which closely resemble the substrate succinate. Okay, so succinate is a substrate on which the enzyme acting is a succinate dehydrogenase, but when we adding the malonate attaching to the active site first, so succinate cannot bind to the active site, so reaction cannot proceed smoothly, so the reaction rate will decrease. This type of the inhibition is used to control the bacterial infection. So for example, in the case of the bacteria, we can take one example for that, that is a sulfur drug. So if a sulfur drug is actually competitive inhibitor of the folic acid synthesized in bacteria. Sulfur drug compete with para-aminobenzoic acid and hence prevent the next step of synthesizing the folic acid. So the reaction will stop there, so the bacteria cannot remain successively, it will die or it cannot reproduce, so the disease will cure. Okay. In the case of the competitive inhibition, let us take this graph, and in this graph you can see the y-axis and the x-axis. On the y-axis we take the velocity, and on the x-axis we can take the Km value, that is a Michaelis-Menten constant. So reaction is proceeding, and it performing like this without the presence of the enzyme. No enzyme, no inhibitor. We are studying the inhibitor. So here this is the A is without inhibitor, and this is the B having competitive inhibitor. Okay, so here this is the Vmax. When the inhibitor is adding to the medium, the Vmax will reduce. So here the Vmax, the half Vmax, if you take the half Vmax and plotting on the y-x-axis, you can see the Km value is increased. In the case of the inhibitor, when inhibitor is added, the Km value is increased. So I already told, when Km value is more, the enzyme is less effective. When Km value is less, it is more effective. So Km value is increased, so the enzyme become, the activity is decreased or it is inhibited. Okay, so here in the case of the competitive inhibition, Vmax that remaining constant, but the Km value is increased. Okay, for example, the succinate dehydrogenase, and this is the succinic acid, and this is the malonic acid. The malonic acid is attaching to the enzyme, but the substrate, actual substrate succinic acid cannot bind, so the enzyme activity is decreased. And similarly, we can study the non-competitive inhibition. So what happens in the non-competitive inhibition? So in this type of the inhibition, the substrate binded to the active site. Actually it binding to the active site. And when the substrate binding to the, means the non-competitive substrate binding to the active site, it alter the conformation of the, or the structure of the active site. When the active site structure change, the actual substrate cannot bind to the active site. So the enzyme activity will decrease. What happens when competitive inhibitor adding to the medium, the competitive inhibitor binding to the active site, near to the active site, not to the active site, near to the active site, and this active site structure will change, so the actual substrate cannot bind to the active site, so enzyme activity decrease. Here also the graph showing Vmax, the Vmax actually decreasing. the Vmax is decreasing, but Km is constant. Km cannot decrease, but the Vmax is decreasing. So in the competitive inhibitor, Vmax is same and Km value increased, but in the non-competitive inhibition, Vmax is decreased, but the Km value is the same, okay, remaining unchanged. So that is the competitive and non-competitive enzyme inhibition. So one more enzyme, that is the allosteric enzyme. So let us see what happens in the allosteric enzyme. Suppose a metabolic reaction pathway, and in this pathway one of the enzyme, one of the product is inhibiting the previous enzyme, then it is known as the allosteric enzyme. See this reaction, glucose converting into glucose 6-phosphate. Okay, for that a phosphate group is giving by the ATP, ATP is converting into ADP, and phosphate is adding to the glucose, glucose converting into the glucose 6-phosphate. This reaction is catalyzed by hexokinase enzyme, okay, and the glucose 6-phosphate amount is increasing, and this glucose 6-phosphate inhibit the hexokinase, so the reaction cannot proceed. So it halted at a particular time. So here the glucose 6-phosphate is the product, that product inhibit the enzyme hexokinase, and this type of the enzyme is known as the allosteric enzyme, and this type of the inhibition is known as the feedback inhibition. Okay, so it is the example for the feedback inhibition. Another example is that fructose kinase, so phosphofructokinase, that is inhibited by ATP, and another example is threonine deaminase inhibited by isoleucine. So that is the allosteric enzyme and competitive and non-competitive inhibition. Let us study a new concept in enzymology, that is the cofactors. So what is actually the cofactors? You know, enzymes are composed of proteins, but there are some other part that is not a protein. So enzyme having protein part and a non-protein part. Okay, and this non-protein part is known as the cofactor. See, enzyme having protein and non-protein part. The non-protein part is known as the cofactor. Okay, and the protein part is known as the apoenzyme. And this apoenzyme plus cofactor, that is known as a holoenzyme. So apoenzyme is the protein part, and a non-protein part cofactor. So apoenzyme plus cofactor, that is known as the holoenzyme. Okay, the cofactor that make the enzyme active. If a cofactor is not there, the enzyme cannot become active. Okay, the cofactors are three types. What are the three types? One of the cofactor is the prosthetic group, another one is the coenzyme, and the third one is the metal ions. Okay, so what is the prosthetic group? It is a cofactor that is organic group tightly bound to the apoenzyme. Okay, it is very tightly organic and very tightly bound to the apoenzyme, then it is known as a prosthetic group. Okay, for example, the reaction 2H2O to 2H2O2 gives 2H2O plus oxygen. Okay, here the peroxidase and the catalyze enzyme is helping to increase the speed of the reaction, or it acting as a catalyst. Here heme is the prosthetic group in the peroxidase and the catalyze. The reaction catalyzing the peroxidase H2O2 to H2O plus oxygen. Here peroxidase and catalyze, both enzyme having a prosthetic group. That prosthetic group is a heme. So heme is the prosthetic group in peroxidase and catalyze. Okay, it is the part of the active site. So then what is the coenzyme? It is also organic compounds that bound loosely with the apoenzyme. If tightly bound, that is the prosthetic group. If loosely bound, that is the coenzyme. Okay, and this loose binding taken place only during the course of catalysis. Otherwise it cannot bind to that. So the example of coenzymes are vitamins. Coenzymes are the vitamins, for example, NAD, nicotinamide, adenine dinucleotide, and NADP, nicotinamide, adenine dinucleotide phosphate. So both containing the vitamin group that is known as niacin. Niacin is the vitamin that is present on this coenzyme. Okay. Then the third one is the metal ions. It form a coordination bond with a side chain at the active site and at the same time form a coordination bond with the substrate. So metal ions that form a coordination bond with enzyme and also with a coordination bond with the substrate. Okay. Example, zinc. For proteolytic enzyme activity of the carboxy peptidase enzyme. So it catalyzes the reaction. Actively is lost. The catalytic activity will lose if the cofactors are removed from the enzyme. So cofactor is very important for the enzymatic activity to be taken place very smoothly. So that is about the cofactors.